Glycolysis, as you might expect, is a very important process because it produces ATP molecules, the energy molecules used by the cell. Now, glycolysis actually has another important function. It is not only used to produce ATP molecules, but it is also used to produce the building blocks of our cells. So molecules such as fatty acids and amino acids. And we'll focus on that in a future lecture. So ultimately, what I'd like to focus on in this lecture is skeletal muscle cells and how glycolysis in skeletal muscle cells is actually regulated. Well, what's the entire point of using glycolysis in skeletal muscle cells? Well, skeletal muscle cells basically want to contract skeletal muscle tissue, and to contract those actin myosin filaments, what must happen is we have to have ATP. And so the predominant role that glycolysis plays in skeletal muscle cells is to actually generate those ATP molecules. But how exactly is glycolysis actually regulated by these skeletal muscle cells? This will be the focus of this lecture. So there are three regulatory points in glycolysis, and these three regulatory points are actually enzymes. What enzymes? Well, phosphofructokinase, hexokinase, and pyruvate kinase. Now, the question might be, if we have so many different enzymes, we have 10 enzymes in glycolysis, why is it that these are the enzymes that are used to regulate the process of glycolysis? Well, because unlike the other enzymes, these three enzymes are the most important enzymes. Why? Well, because they essentially regulate virtually irreversible processes that only take place in one direction. So let's take a look at phosphofructokinase. This enzyme catalyzes step 3, and this happens to be the most important enzyme. Why? Well, because it regulates the commitment step of glycolysis. So it transforms fructose 6-phosphate into fructose 1,6-bisphosphate. When we're at this stage, the fructose 6-phosphate, before it has been transformed, it has a choice. It can either move into the active site of the phosphofructokinase, in which case it will be transformed into this product, or it can go on to form glycogen. But once we form the fructose 1,6-bisphosphate, this product has now been committed to the process of glycolysis. So this is the most important enzyme because it essentially tells this molecule to continue and commit to that process of glycolysis, and there is no turning back for this molecule. Once it's committed, it has to carry out the process of glycolysis. So that is why this enzyme is the most important regulation site in our cells in the process of glycolysis. It catalyzes that commitment step. Now, if we study the structure of phosphofructokinase, we're going to find catalytic regions and regulatory regions. The catalytic regions basically contain the active site that binds this substrate, fructose 6-phosphate, but those regulatory domains contain the sites that an allosteric molecule or molecules can actually bind to. What are these molecules? Well, before we look at these molecules, let's define something called the energy charge of a cell. The energy charge of a cell is basically the ratio of ATP molecules to AMP molecules. So if the energy charge of the cell is high, that means we have a high ATP value relative to AMP. If the energy value is low, we have a low ATP ratio relative to AMP. And these are the two allosteric regulatory molecules of phosphofructokinase. So let's suppose we're at rest. And what that means is our muscles aren't actually contracting, and so our muscles do not require ATP. And there will be a buildup of ATP in our cells, and that means the energy charge value will be high, because if the energy charge value is high, that means ATP to AMP ratio will be high. So what happens is these excess ATP molecules move on and bind onto the regulatory sites of phosphofructokinase, and what they do is they essentially decrease the affinity of phosphofructokinase for the substrate molecule, the fructose 6-phosphate. And so what happens is step 3 basically stops. taking place. And if step 3 stops taking place, then we're not going to produce the ATP molecules. And that makes sense because if we're not exercising, if we're at rest, we don't want to produce ATP molecules. And so that turns off the process of glycolysis. But what happens if the energy charge value is low? So in the case of us exercising, what happens is the ratio of ATP to AMP decreases, and what that means is we want to produce those ATP molecules because we want to be able to contract our muscles quickly and effectively. And so what happens is the AMP goes on and binds onto the regulatory sites of phosphofructokinase, and that stimulates the activity of the enzyme to catalyze this process, increase the rate of the process, and increase the rate at which the ATP molecules are ultimately produced by glycolysis. But the question is, in the process of glycolysis, we only find ATP molecules and ADP molecules. AMP is not found in glycolysis. So why is it that AMP and not ADP is that regulatory activator molecule for phosphofructokinase? Well, because something actually happens when we have a very low concentration of ATP inside our cells. Instead of using glycolysis, our cell does something else. So to basically be able to quickly produce the ATP molecules that are needed by the cell to basically contract those skeletal muscle tissue, this is a process that takes place, and it's catalyzed by adenylyl kinase. So adenylyl kinase takes 2 ADP molecules, 2 adenosine diphosphate molecules, transfers 1 phosphoryl group from this onto the other molecule that produces very quickly an ATP molecule and an AMP molecule. And these ATP molecules can be quickly taken by the cell to basically contract those skeletal muscle tissues. And we also produce this AMP molecule, and it's this AMP molecule that increases in concentration and that decreases the energy charge ratio, and that causes the binding of the AMP onto that phosphofructokinase to stimulate it to produce many more ATP molecules via the process of glycolysis. So under low ATP conditions, the cell will use adenylyl kinase to create ATP from 2 ADP molecules in a process that generates AMP and ATP relatively quickly, and that allows the cells to basically continue contracting those actin myosin filaments. So let's summarize our result in the following overview, in the following graph. So we have the fructose 6-phosphate catalyzed by the phosphofructokinase to produce this molecule not shown here. Then some number of processes take place, and ultimately we produce the ATP molecules and pyruvate. Now, when we're actually not exercising, when we have lots of ATP molecules, we don't want to produce the ATP molecules, so they essentially go on and inhibit the activity of phosphofructokinase. So we see that ATP is an allosteric inhibitor of phosphofructokinase, but AMP is an allosteric activator. Now, actually, something else also affects the activity of phosphofructokinase, and that is the pH inside that tissue. So if there is a drop in pH, what that means is there's an increase in the H+ ion concentration. So why would we find a higher H+ concentration in exercising muscle cells? Well, when muscle cells are exercising, sometimes there is not enough oxygen to actually go around those cells. And so to basically continue producing those NAD+ molecules, our cells begin lactic acid fermentation. And when lactic acid fermentation takes place, we produce a lactic acid ion and a lactic acid molecule, and that quickly dissociates into lactic acid and releasing that H+ ion. And so as the fermentation process takes place, it increases the H+ concentration, that decreases the pH, increasing the acidity, and that can cause damage to that exercising muscle tissue. And so what our cell does is, to prevent damage due to that acidity, our glycolysis process is shut down to prevent any further production of those H+ ions. And so we see a low pH can actually inhibit the activity of the phosphofructokinase. This is because as the cells rush to meet the high ATP demands, fermentation produces lactic acid, which dissociates into H+ ions and lactate ions, and that increases the acidity, drives the pH down, and that can cause damage to that tissue. And so to prevent this from happening, the glycolytic cycle is essentially turned off by inhibiting the phosphofructokinase. Now, let's move on to hexokinase, and let's recall the first three processes in glycolysis. So in step 1, we have glucose transformed into glucose 6-phosphate by hexokinase. This is also an irreversible process. We have a reversible process that is catalyzed by phosphoglucose isomerase. We transform glucose 6-phosphate into its isomer fructose 6-phosphate, and then this third step takes place, in which we transform the fructose 6-phosphate into this committed molecule fructose 1,6-bisphosphate by phosphofructokinase. So let's suppose we're at rest, and what that means is we're going to have high energy charge value. We're going to have many ATP molecules. What will that cause? Well, the ATP molecules will bind onto the regulatory sites of phosphofructokinase, inhibiting that molecule. And if this molecule is inhibited, if the enzyme is inhibited, this fructose 6-phosphate cannot go on to the next molecule. And so there will be a buildup in the concentration of fructose 6-phosphates in our cell. Now, fructose 6-phosphate is in equilibrium with glucose 6-phosphate, and by Le Chatelier's principle, if we increase the concentration of this product, we're going to increase the concentration of this reactant as well. So the increase in concentration of this increases the concentration of the glucose 6-phosphate, and when that takes place, the rise in the concentration of glucose 6-phosphate causes a negative feedback loop with the hexokinase, and so it goes on and binds onto hexokinase, and that causes hexokinase to basically inhibit its activity. And so we see that via the increase in the concentrations of these two molecules, the phosphofructokinase communicates to the hexokinase to basically stop carrying out its function, to basically stop the process of glycolysis. So they work together to basically stop producing ATP molecules when we have plenty of ATP molecules in our cells. So once again, hexokinase catalyzes the conversion of glucose into glucose 6-phosphate. And as the ATP level rises in the cell, phosphofructokinase is disabled, and this causes the buildup of the fructose 6-phosphate. And because fructose 6-phosphate is in equilibrium with glucose 6-phosphate, that causes a rise in the concentration of glucose 6-phosphate. And this rise in glucose 6-phosphate creates a negative feedback loop that essentially inhibits the activity of the hexokinase. And when this takes place, the cell essentially stops uptaking the glucose from that blood, from that nearby environment. And so when the hexokinase is turned off, the glucose will remain in the blood in that surrounding environment. And in this manner, we see that the inhibition of the phosphofructokinase by the ATP will ultimately inhibit the activity of the hexokinase. And finally, let's move on to the pyruvate kinase. So pyruvate kinase is the enzyme that catalyzes the last process in glycolysis, and this is also virtually irreversible. It only takes place in this direction. So we have phosphoenolpyruvate that is transformed into pyruvate molecules and ATP molecules by the activity of pyruvate kinase. Now, just like phosphofructokinase is inhibited by ATP molecules, so is pyruvate kinase. So when there's a rise in ATP, when we have a high energy charge value in the cell, when we're not exercising, we have too many ATPs, And so the ATP acts as a signal to basically inhibit the activity of pyruvate kinase. It creates a negative feedback loop, an inhibition loop. Now, as I mentioned in the beginning, glycolysis is not only used to actually create ATP molecules, it's also used to produce building blocks such as amino acids. And so, there's no surprise that alanine is also an inhibitor to pyruvate kinase, because pyruvate is actually used to produce alanine. And so if there is an increase in the amount of alanine that we have in our cells, if we have too much alanine, then we don't want to produce the pyruvate anymore. And so the alanine will go on and inhibit the activity of pyruvate kinase. The final thing I'd like to discuss is what happens if we have low concentrations of ATP. How will that affect the activity of pyruvate kinase? Well, when we have a low amount of ATP, what that means is we'll essentially have the AMP binding to the phosphofructokinase, stimulating the production of fructose 1,6-bisphosphate. And fructose 1,6-bisphosphate will create a positive feedback loop with the pyruvate kinase.